All right, folks, on to part 3 here. We just finished up laying down primase, so I've moved on to the next slide here, and I apologize for the length of these videos, but they are very informative. There's a lot of information here, so I'm trying to save you some time, actually. So ultimately, what we have to remind ourselves of is we've got our helicase over here, we've got our topoisomerase over here, and we've got our primases here that, and I need to redraw them, but our primases have laid down strips of starter strip RNA going in opposite directions of the template strand. Remember that they're going to lay from 5' to 3' on both sides. So 5' to 3'. And these are new starter strips of RNA. So the RNA is going to be pairing up with the DNA on the opposite template side. Remember that template was kind of exposed, right? And they lay down about 5 to 10 pieces. Next thing that comes in is going to be an enzyme, a very important enzyme called DNA polymerase 3. DNA polymerase 3. It's abbreviated DNA pol 3, and that's the way I'll describe it. DNA pol 3 finds the end of the primer. So this primer stops at some point, and then pol 3 says, oh, okay, this is where I pick up. This is where I start. And so what pol 3 does, and there's going to be multiple of them working on the same strand here, or on opposite sides, DNA pol 3 is going to find that primer, and it's going to continue from that point. It is going to lay down DNA. It's going to lay down the correct DNA. So I'm going to use blue because we're going to indicate that that's the new DNA that's being put in here. So this new DNA is going to be laid down, and this pol 3 is going to work in this direction again. It's going to work still continuously in the 5' to 3' direction. So this thing here is going to go, and it's going to travel down. Where'd my pointer go? Where'd my cursor go? Where is it? There it is. It's going to travel down, and it's going to continue working in this direction all the way to the replication fork. So I'm just going to go ahead and draw that down there. So I'm going to show that this moves in this direction all the way down here. So DNA pol 3, again, is going to be moving from 5' to 3', and it's going to move all the way down to the replication fork. This one's going to do the same exact thing. It's going to move all the way down to the replication fork. So it's just going to keep like zipping along. It's going to keep inserting the correct DNA pairings as it goes along, as it navigates down. Now, it gets down to the replication fork, and you have to kind of animate this thing in your head. You have to understand that what's going on here is this helicase, as this thing is moving along, as polymerase is moving along, helicase is moving along too. And it's pushing, in this direction, and to the left over here and to the right over here, and it's opening up more DNA. So overall, the whole thing is, working together, all in unison, all at the same time here. And the whole thing is moving in that direction. And this whole thing is moving in this direction. And the topoisomerase is releasing stress and blah, blah, blah. Hopefully you can animate some of that in your mind. Now, we've covered only one side here, though. What about, these sides here? There's going to be a problem that we encounter here. But so far, this is what we need to know, okay? So moving on to the next step, you can pause it here and get caught up. But I'm going to move on to the next step here in a second. All right, so once again, writing down my thing here, the primer is going to be laid down here, right? And then our new DNA is going to be laid down from here to here, moving towards the replication fork. And the same thing is going to be happening on the other side here. Primer was laid down, and new DNA was started here and continued in this direction by pol 3. The next step is another enzyme called DNA polymerase 1. What DNA polymerase 1 does, and you want to write it down, it's a spell checker. It's a spell checker. It basically goes through the DNA strand and it identifies where there's RNA, and it says, wait a second, you shouldn't be here. You got to be DNA. So it swaps out the RNA, so it's going to target these primers. It swaps out the RNA, and it says, well, I'm going to replace you and spell check you, basically. I'm going to make sure that you're the right stuff. So what pol 1 does, what polymerase 1 does, is it's going to hop on board here, and it's going to hop on board any primer that it finds, and it's going to replace that primer with the correct DNA now. And guess what direction it works in again? Always works in 5' to 3'. This is going to cause a problem. So it's going to replace these things. So it's going to chuck out the RNA. It's going to say, hey, you don't belong here. It's going to throw the RNA out. So it literally ejects it out of there, and it replaces it with pieces of DNA. So it puts in the right A's, T's, C's, and G's. So it proofreads. So it fixes a problem. But there's another problem that's occurred here. It only goes to the end of where the primer was, but it can't glue the gap together. So there's a gap that's going to be right here. There's going to be a gap that's right here. And there's going to be a gap that's right here between where the primer was and where DNA pol 3 took off. And that gap needs to be fixed too. So that's going to be our next step. So I'll pause there, let you get caught up with notes, but I'll move on to the next frame here. All right, so we've got those gaps that were created. And again, I know I'm kind of redrawing a bunch of stuff here, but this is where the primer was. This is where pol 3 took off from and moved in this direction. This is where the primer was, and remember, it got replaced by DNA pol 1. Replaced it with DNA instead of RNA. And now an enzyme called ligase is going to come along, and ligase is going to take these ends here, and it's going to glue them together. So it's going to identify little gaps that exist, and it's going to take those, and it's going to glue them together. It's going to make a bond occur kind of magically between there. It's not magic, it's chemistry. But it's going to put that all together into one continuous strand then. So that's ligase's job, is to glue together the gaps that exist between where the old primers used to be and where DNA pol 3 took off from. So again, putting this kind of all in motion in your head. We had our DNA, we have helicase, we got topoisomerase, primase comes in, sets a primer, DNA pol 3 comes in and says, hey, there's a primer, I'll go ahead and start there. It clicks along. DNA pol 1 comes through and says, hey, primers, you're in the wrong place, flips them out with the DNA for RNA, so swaps those out correctly, and then ligase comes along and glues all the gaps. Okay, so hopefully you're getting the idea here that what we're ultimately creating here is new DNA that is, this is a new strand of DNA being created up here. And this is a new strand of DNA being created down here. All right, so all of this is being created. Now, so what about these other spots? There's going to be a problem. Okay, so I'll do it this way. I'm going to split this thing this way. Now, not all diagrams are the same because my 5' and 3' are specifically drawn the way that they were. In my diagram, we had a primer that got replaced, and then the whole thing just worked in this direction, and it continued towards the replication fork. We had a primer that was in this direction, and then the whole thing continued towards the replication fork. So it can go continuously, all the way to the very end of the whole bubble, all the way to the end of the DNA if it needs to. But what about these other spots? Like, how does it catch up? Because what happens here is that as this part of the whole process keeps getting pulled apart, the helicase, which I don't have drawn here, helicase keeps zipping that thing apart and exposing more stuff. A primer gets laid down, but keep in mind which way the primer is facing. It's anti-parallel now. It's going the opposite direction. So when it lays down a primer, let's say it lays down a primer right here, it starts right here, the primer actually lays in this direction. But the whole thing is opening up behind it over here as we go along. So it has to it has to hop off, the primase has to hop off of the DNA here. So it used to be right here. It pops off of here and it leaves that primer back behind. But now it's going to go back to here and it's going to lay down another primer strip. It's going to say, well, I need another primer strip here. And then this thing is going to continue to open up some more over here. And another primer strip is going to go in there. And so it's going to continue to lay another little primer strip, and it's going to go the other way. Well, it's kind of the whole idea of like 2 steps forward, 1 step backward type of thing. What it does is it keeps laying primers down, but it has to keep working backwards. So it works 5' to 3', but then it has to take like 2 steps back, then 5' to 3', then 2 steps back, then 5' to 3', and 2 steps back. So it's constantly kind of jumping backwards to keep catching up with the replication fork. Think about what that statement just meant. It keeps jumping backwards to keep catching up with the replication fork. Well, what ends up happening is that when everything fills in here, we have all these little fragments of DNA, all these little segments on the other side of the ladder. So on this side, they'd be going this way. There'd be one little fragment here and one here. And ligase really has to work a lot on the other side. So those little fragments were once discovered. And those fragments are called Okazaki fragments. There's the term right there. So they're like little tiny patches of DNA that are used to catch up with the replication bubble. Because once you get to the middle of the replication bubble, then you can go continuously towards the fork. Okay, think about that. So once you get to the middle of the replication bubble, then you can work continuously towards the replication fork. So ultimately, this thing, this side over here and this side over here, this is called the lagging strand because it takes longer to do this side. This stuff here is called the leading strand. And so is this one over here because it just goes. Now the problem is that most students just kind of like, yeah, I'm going to memorize this. Lagging's top right, leading is top, bottom right. You're doing it wrong because quite honestly, if you think about this here, let's go to here. If I had my DNA like this, and I said, all right, here's the 5' end. Well, where do we have our 5'? This would be 5' over here, and this would be 3' in this direction over here. But let's say that a drawing flips it. So they have their little drawing, and they have it coming in, and then they got their little bubble, and they have this, right? They get their little bubble. Let's say that they flip it on you. They say that this is 3' and this is 5'. Well, now this is 5' over here. This is 3' over here. This is where you guys get confused, but think about it. So we have our 3' over here. That means new DNA that gets built is going to be built in this direction. And so this is now the leading strand. This is the leading strand. And then these are the lagging strands going back this way. So if you try to memorize a pattern that basically gets you to think like, oh, top right is lagging, bottom right is leading. No, it doesn't work that way. Okay, so you got to think about the 5' and 3' stuff and which way it leads. Okay, so moving on to a checkpoint. Here you go. Can you put the following order? Blah, blah, blah. I'll pause for a second, then I'll answer them. All right, so hopefully you guys can answer these. I threw this little meme in here. Hopefully you get a little kick out of that. The following is going to be what comes first. Helicase comes first, and then topoisomerase relieves the pressure, and then pull 3 comes in, and then pull 1 comes in, and then, I'm sorry, I forgot about, I skipped over primase here. So helicase, topoisomerase, primase, pull 3, pull 1, ligase. Sorry, I messed that up. Ultimately, you should be familiar with them. Pull 3 and pull 1 are confusing because pull 3 happens first. And again, that has to do with when they were discovered. Pull 1 was discovered first, and then pull 3 was discovered later. And they say, well, it's pull 2 was actually discovered later. And then pull 3 was discovered later. And then they realized, like, in order, they actually work in different order. So sorry, we don't rename them. Can you name the primary functions? So helicase, unwind and unzip. Topoisomerase, relieve stress. Primase, lay down RNA primer. Pull 3, lay down DNA, pick up where primase left off. Pull 1, spell check and replace RNA with DNA. And then ligase to glue the ends together. Okay. Which direction do all enzymes work? They work in a 5' to 3' direction. They build in that direction. The template strip is opposite, remember. So the template strip is opposite, but where they're building, they build from 5' to 3'. They build new stuff 5' to 3'. That's the specific part. Difference between leading and lagging strands. Leading strands go really quickly. Lagging strands go very slowly because the primer and the DNA and everything, all the enzymes are working away from the replication fork. That's the key. They work away from the replication fork. And so they have to build it in small little segments called Okazaki fragments. Okay. And then the answer to number 5 is duh, obviously. All right. I'll leave it there, and there'll be one more little piece here, probably 5 minutes long. So meet back for part 4. Thank you.